right, welcome everybody to the Oncology Researcher Roundtable. I'm really excited to talk about cancer research today, and we have an amazing panel with us. Yeah, I really want to introduce our panel today. This is going to be amazing. So first, I'd like to introduce Yin Cao from WashU in St. Louis. Yin runs an amazing group of folks running a computational lab interested in early onset cancers. And I mean, 2 days after the proteomic data came out on UKB, I saw some preliminary data from her lab, and we're going to talk about that, and that was awesome. Also, Kezia Geitskel, she's an epidemiologist, does a lot of different stuff. She's going to tell you all about that when we go through and introduce everybody. Megan Conroy from the UK Biobank, who's very interested in oncology and knows a ton about the UK Biobank. Alex Bick, who told me earlier that he sequences meerkats as part of his job from the Boston Zoo. So that's great. I'm Ben Busby. I'm from DNA Nexus. But right now, I'm going to turn it over to my colleague, Brenton Pyle, who's going to go over logistics, tell you about a few other things you might need to know about the rep. And then we'll go ahead and get started and introduce our panelists and talk a whole lot about oncology research. So, Brenton, over to you.
Thank you, Ben. If you have a question during the webinar, you can drop that in the Ask Questions section. That's the module. You can find that at the bottom of your screen. It has a question mark box over it and is kind of an orange color, and we'll be able to answer that during the webinar, and also we'll be able to save some for the end and answer them live. I also have some few updates and related things that you might be interested in if you're interested in the research analysis platform. Ben mentioned the community.dnanexus.com. That is the community forum for UK Biobank researchers to collaborate and also access other helpful documents, sample pipelines and things. There's tutorials and tools shared by the community that that has helped a lot of people get the most out of working on the research analysis platform. You can find the sign up link for that in the related content section or scan that QR code there. I'll be mentioning the related content section a couple times. That is the paperclip icon at the bottom of your screen. If you are an early career or a researcher from a low or low middle income country, you can actually get funds to work directly on the research analysis platform. You are also entitled to reduced access fees to the tier 3 data. That's the large data, the genomic data you'll be able to access there. You can apply, again, through the related content link. I'll hide that one more time, or scan there to be able to get either kind of getting started funds to just start working on the platform, or enhancements to work on an already active project. If you have been working on the RAP and are looking to collaborate to develop more tools and navigate the rich data to kind of get your projects up and running, we have the Research Analysis Platform Accelerator. This is working with the DNA Nexus Xvantage team to get the most out of there, and they work with commercial orgs to develop tools and help assist with anything that you might need for your research project. Again, this is also found in the related content. You can ask for more information, scan that QR code, follow that short link to get in contact with us, and we can provide more information on those packages. If you're part of the community and you would like to promote your research, we're planning tons of events for the future. So we would like to hear from you. So if you have anything that you'd like to present, highlight in our monthly newsletter spotlight, organize a meetup or workshop, please let us know through that bit.ly link, also found in the related content section, or scanning that QR code. And our team will kind of help assist with planning and organizing whatever makes sense for what you'd like to do, whether it's, again, the webinars, research roundtables, or just highlight in the newsletter spotlight. We want to promote the community and collaborate with researchers that are doing amazing things. So please let us know. And finally, if you are not on the platform already, not part of the community, but you're an approved UK Biobank researcher, it's part of your approved application to join the research analysis platform. It is free to join, and you even receive £40 credit just for signing up, and that's to get you started working on the platform. So you can register at that bit.ly link there, related content section, and scan the QR code as well. And again, if you're an approved UK Biobank researcher, you can join immediately, and it only takes a few minutes to sign up. All right, that is it for me. Ben, I will pass it over to you to kick off the webinar.
Fantastic. All right. So I think what we want to do is really get sort of an understanding of why the participants are interested and motivated by cancer research. So let's go participant by participant and talk about really why you're interested in cancer research. So maybe we can start with Kezia, and she just happened to pop up on the screen. So that's awesome. So Kezia, how did you get interested in cancer research? Why do you still do cancer research?
So I think a lot of my interest in cancer research really comes from my clinical work. So I spend part of my week as a clinical pathologist, which means I mostly look down a microscope and try to work out whether or not people have cancer. And if they do, then I try to determine other information that is important for prognosis or for planning further treatment. So what kind of cancer do they have? How far is it spread? Does it express particular molecular markers that might affect treatment? And I think if you do that, then it automatically leads to bigger questions around why do some people get particular kinds of cancers? Are there specific risk factors for particular types of cancer? Are some of them modifiable? And so I think, as with most people who do cancer research, I have this hope, which I think is well-founded, that if we come to understand how and why different types of cancers develop, then that will enable us to devise better strategies for early diagnosis and prevention and treatment. And so because I'm a pathologist, I'm particularly interested in trying to sort of drill down a bit more into saying, okay, it's not just breast cancer. There are lots of different types of breast cancer. It's not just ovarian cancer. There are lots of different types. They look different down a microscope. They express different molecular markers. They probably have different risk factors. And we've got some evidence that they do. And so that's why I'm really interested in sort of big picture epidemiology to try and link our sort of biology understanding of the sort of microscopic pathology and the molecular changes with our big picture epidemiology.
That's awesome. So Kezia, if I can ask you to go a little farther, and then we'll get to other participants. I mean, cancer subtyping is hot right now, right? Particularly for small cell lung, ovarian, colorectal, a couple other types. How do you actually address that as a pathologist, right? Like we have like molecular subtypes and talk about that all the time, but actually in the lab as a pathologist, how do you think about subtyping?
Well, that's a really interesting question. You'll have to stop me at some point because I get overexcited about this.
Let's go with 40 seconds.
So basically, our classification systems keep changing in pathology. Literally, we rewrite the books every few years as our knowledge improves. So, 50 years ago, your classification system was a pathologist with a pink and purple hematoxylin and eosin slide and a microscope. And pathology reports from 50 years ago are so short, because there's very little information that's going to make a difference. Whereas now, I don't just diagnose an endometrial cancer. I need to say, does it have P53 mutant expression? Does it have mismatch repair loss? I might send it off for POL-E testing. There are so many different molecular tests that we have to do. So yes, I will give the old classification of what kind of a type does it look like down a microscope, but then I need to give all the molecular information because that's now governing the way that we treat. And it's only going to get more detailed. Every time we come up with a new molecular piece of information that we know makes a difference either to prognosis or treatment, we suddenly have to rewrite all our pathology reports.
That is fantastic and really seeds for future discussion. But I want to introduce the rest of the panelists. Also, I mean, if you've never considered political debate, I mean, it'd be amazing because like you have these like other interesting clauses and sentences. But so let me jump over to Yin Cao because I think this follows on nicely with Yin's work on early onset cancers and thinking about how those are really different than some of the canonical presentations you have with folks of sort of the peak of the bell curve. Yin, are you there?
So a really nice transition to my interest. In the past several years, we have been really fascinated by the rising instance of multiple early onset cancers, not only in the United States, but also globally. So we tried to leverage different resources, usually very large database, including large cohorts, such as the Harvard-based cohort, and also UK Biobank for some of these initial and rapid discovery in terms of trying to identify putative as well as emerging risk factors for early onset cancer. And this is really becoming one of the concerns, not only in the United States, but also globally. We saw an increase in multiple, especially gastrointestinal cancers and also other cancers in younger population. And we saw a clearly described first cohort effect, which is very interesting. We are still trying to find the clues, but I think right now there are pieces of information either at the risk factor level throughout the life course and also molecular level, either using molecular subtyping or potentially proteomics markers, etc., metabolites. We are trying to bring these pieces of information together. I think overall, this is really a fascinating area to me and really interests me because it has broader impact to the global population and really help with cancer prevention down the road for our younger generations.
Thanks, Yin.
So that's my interest.
Thank you. So moving now to Alex Bick. Alex, I know you work on clonal hematopoiesis, which is of great interest to lots of people. Alex, can you tell us a little bit about clonal hematopoiesis and also really how that predisposes specific cancer population?
This one of the things we've been really interested in is this notion that our DNA changes over time, and everyone in the cancer audience says, yeah, we know that that's what causes cancer. But I think until we started sequencing these really, really large biobanks, such as UK Biobank, we didn't have an appreciation of just how common this somatic mutation process was. And so in the blood, when our blood stem cells acquire a mutation that gives that blood stem cell a proliferative advantage, you might have a clone that starts taking over 5, 10, 20% of your blood, all coming from this one mutated blood stem cell. And so when we started sequencing large numbers of people, we found that this was quite common. That 10, 20% of people, once they get into their 70s and 80s, have one or more of these very large clones floating around in their blood. And initially, we were really annoyed by them because they were sequencing artifacts that were really getting in the way of analyses that we were trying to do of germline genetics. But then over the past, I'd say 5 years, we've actually embraced them and realized that this is a really important pre-malignant state, and that what we're learning from it teaches us not just about this precancerous condition, but also shares some of the same genes and pathways as other kinds of blood cancer. And so in the past, we've had small cancer data sets, perhaps 10,000 cancer genomes. But with clonal hematopoiesis, we actually have hundreds of thousands of precancer genomes and can learn many of the same things.
Awesome. So Megan, Alex, and Yin have mentioned the UK Biobank specifically, which I was planning to ask them about a fair amount in the rest of the webinar. But can you talk a little bit about other reasons that Yin and Alex haven't mentioned that the UK Biobank is really useful for cancer research?
Certainly. Well, I think UK Biobank's biggest strength is just the sheer amount of data that we have. So our participants, they answered, I think, a 3.5-hour questionnaire when they first came along to our assessment centres on so many different lifestyle factors and diet and exercise, job histories, medical histories, you name it, we asked it, and they all kindly provided this information. But going beyond that, we have, and we still do, enhance the data sets as much as we can. So we have accelerometer data, objectively measured physical activity on 100,000. We have linked to the hospital data, the cancer data, the death data. for all of our participants. And then we also have primary care data as well for about 40% of them. So we just have so much data as well as the genetics. And then there's some imaging. And so you really can answer, there's so many questions that you can look at in relation to cancer, which just wouldn't be possible without this kind of vast amount of data that we have.
So we had a proteomics webinar, a proteomics on the UK Biobank webinar a couple of weeks ago, and folks can find it at community.dnanexus.com if they're interested. But what I'd ask you is, that really kind of went viral. Like, I mean, hundreds, thousands of people like signed up for it and so on. And so why do you think that went so viral? Why are people so interested in proteomics? And I think we'll cycle back to the rest of the folks to ask them why they're interested in proteomics.
So proteomics is very new to me. It's not something that I have delved into yet. But I think it's just that understanding, genetics is shown us so much, but actually how does the genes lead to the proteins, and how do the proteins lead to disease? And really being able to dig into what's going on there. Is it protein folding rather than genetics? Why does the genetics always not lead to disease? And why, what's going on there? It's that kind of middle step in between the kind of genetics and disease. And it's kind of the first time we've been able to do that within this kind of sheer size, and hopefully more and more data will become available for that.
So thank you for that lovely introduction and allowing me to ambush you with that question. And so I'd like to open this up to the rest of the panel. I know I mentioned Yin is looking at the proteomic data, but I'm curious for everybody on the panel, how do you think the proteomic data is really shaping oncology research, and how are you using the proteomic data in your research? And please feel free, anybody, to just jump in. All right. With this webinar, I'm going to call on specific people. So, Yin, can you tell me a little bit?
Let Yin go first.
Oh, okay, cool.
Yeah, I think I totally agree with Megan. Proteins are really central in terms of disease, and we really I think our understanding of the proteome is really limited by the technology a couple of years ago. But now with this new platform from Olink, which enables us to look into thousands of proteins at the same time. And what is really unique about the UK Biobank is also that because of its large scale, it's kind of agnostic in terms of disease phenotype. So, which means that in the cancer space, we can potentially identify shared markers, protein markers, across different cancer type. We can also compare markers for particular, whether the marker is specific for a particular cancer. And because of the large-scale profiling that were provided by the UK Biobank, such comparison become possible. Whereas in many real-world research settings, we have to do such profiling, disease by disease, cancer by cancer, which really limited in terms of the time and how we can push forward rapid discovery, especially in the context of the next steps in terms of identifying the potential drug target and gene-protein interaction, protein-protein interaction, etc. So I think that really speaks to the advantage of leveraging UK Biobank for proteomics analysis in the cancer space.
Awesome. Alex, I think you wanted to add something there, if I remember correctly.
I think what's really exciting about proteomics is that from an intuitive standpoint, it feels more interpretable. And I think the webinar that you had a few weeks ago was sort of emphasizing that it's actually not that simple. But sometimes when we're doing genetic analyses, we find a genetic association between a germline mutation, variant, and some trait, whether it's cancer or something else. But often these variants are not in the coding region. They're affecting one or multiple genes through some regulatory mechanism. And so it can be a lot harder to say, what is it that we've actually found? Whereas with the protein, we think that we're measuring a circulating protein. And I think the webinar emphasized that is often the case, but actually is more nuanced based on how the proteins are actually being measured. So I think there's also a lot of interest because they're also great therapeutic targets. If indeed the circulating protein is causal, then there's easy ways to modulate that, perhaps through a therapeutic antibody.
Awesome. So if we get to it later, I'm going to ask you about TCGA and VU and other databases and bridging the gap. But I want to get to something else first, which is an audience question. And I think this is really pertinent to what you and Yin said. How do you define an appropriate cohort population in a study aiming to find biomarkers for early cancer diagnosis? I have some ideas, but I'd really like to hear Yin, Alex, Kezia, any of you want to jump in on that?
I think that's a really great question. From epidemiological perspective, we usually, when we look into early biomarkers for early cancer diagnosis, we usually confine to 1 year before diagnosis. Along that timeline, basically the control population will be individual without cancer during that time. And if we are more interested in etiological markers that potentially will predict the risk of disease, we'll go back more in time, looking into more like at least 1 year above before diagnosis, in terms of trying to identify biomarker during that time window compared to controls who are matched in the sense that they don't have cancer when the case were sampled. So that's pure epidemiology perspective. And I'm open to hear to what the other panelists would like to share.
I think one of the things that is important with UK Biobank in particular is having a really solid understanding of who these people are and how they made their way into the study. So one of the things that we've noticed, both in cancer and cardiovascular disease and others, is that UK Biobank participants are actually much healthier than the average person is in the UK. And also there tends to be quite a bit of relatedness. That there's some large families that sort of everybody signed up, which is wonderful for certain genetic reasons, but can sometimes make it hard to do some of the epidemiology analyses that Ian was mentioning. And so just knowing that people had to live to 40 years old before they signed up for this study means that if you had an early cancer in your 20s, let's say it was caused by a strong cancer predisposition gene like BRCA, some of those patients may actually have died and never been able to sign up for the UK Biobank. And so particularly for cancer, it's been very interesting to see some of these features come up, making control populations even harder.
Awesome. So I'd like to sort of switch over from controls, although if I may take the moderator's prerogative for 5 seconds and say having multiple control populations is awesome for statistics. So if anybody puts some strategies for that on GitHub, I will buy you a beverage of your choice. That said, I'd like to sort of switch over to future data sets and ask Megan and Kezia. I mean, we know that the single cell data is coming out. And so how do you think the single cell data is going to affect these kinds of analysis when that comes out in the spring? How does that play off the proteomics data?
Well, I'll give it a go. I guess it's your first step in your transcriptomics. So it's just another piece of information there to look at, and I get there'll be researchers out there that are going to be doing some amazing things using all of that data that's available, and then there'll be others who are doing other amazing things, just looking at the single cell data itself. But it's another step in the pathway, and to be able to really understand what's going on and what's causing disease in these different populations.
So, Kezia, I know it's really not appropriate to ask people to predict the future, and so I won't ask you about timelines, but I'm wondering, how you see things from, say, single cell data or proteomics entering the clinic in terms of like subtype diagnosis?
It's a really interesting question. So I think for the proteomics, I mean, I guess there's kind of two ways I think about it. One is around, can we do some analyses, or sort of from a clinical diagnostic perspective, can we do things without having to get the sorts of samples I normally get? So generally, for me to diagnose cancer, someone needs to go into the patient and get a bit of tissue. And if it's, if it's something on your skin, that's relatively trivial. But if the thing that we're worried about is in the depths of your body because it's in your pancreas or somewhere in your liver, then trying to get a biopsy is much more awkward. And so if we could find ways of diagnosing cancer accurately, getting the kinds of information that we need for prognosis and treatment without having to get biopsies in cases where it's going to be difficult, that would obviously have clinical advantages. And so there are people exploring lots of different strategies for that. So some of the audience will have probably heard about the idea of liquid biopsies. And often that is trying to look at sort of small amounts of DNA that might be tumor DNA in the blood. And so there are various sort of other research projects around people trying to think about earlier diagnosis of cancer on the basis of sort of cell-free tumor DNA. But, or in some cases, even thinking about small tumor cells in the blood. But I think you can also sort of take an approach, and some people are, of saying, are there proteins in the blood that might be detectable by the proteomic assays that we're using in UK Biobank, and might some of those be sufficiently specific that we can either get a diagnosis that is good enough for us to do treatment, though I think we're quite some way away from getting the kinds of information we would really currently get from pathology. But it might be enough information to help direct people down sort of different investigative and treatment pathways on the basis of those kinds of proteomic signatures, if we can get better at interpreting them. Obviously, I still want to do a plug for the value of tissue-based pathology, but I think there is a huge amount of sort of potential for non-tissue-based forms of cancer diagnosis. Even if it's not the only form of cancer diagnosis that you get from that patient, it might be the sorts of things where we can get useful information earlier on in the process to try and speed up our diagnosis and treatment. So I think there is really exciting potential, but we're not there yet.
So Kezia, that's awesome. If I may ask you another difficult question, and I'm really hoping that you don't punch me at the UK Biobank Symposium in December. But what I'm wondering is really, how do you think about taking results from your own clinic and introducing, like really integrating them with UK Biobank results and say results from other things like TCGA, like, I mean, they have CPTAC and all that. I mean, how do you think about making that data work together?
So I guess there's several approaches. So one would just be the usual thing that we try to do in medical research, cancer epidemiology, around sort of triangulating and comparing results from different data sets. And so ask the same question or very similar questions in data sets which may have slightly different populations or slightly different availability of data. And so there's an extent to which we can already try to do that with things like TCGA and UK Biobank, and sort of seeing are the associations that we see consistent across these populations, and does that play out in our clinic data? And so we can already do that, and it's valuable. Just as a plug for some of the things I'm currently really excited about with UK Biobank, because obviously I think pathology is really exciting. One of the things that we're trying to pilot is taking some of the glass pathology slides from some of our UK Biobank participants who've been diagnosed with cancer, and scanning them to make digital pathology slides. So we've been doing that at the moment. I've already got my first couple of hundred colorectal cancer pathology slides from UK Biobank that we've scanned, and we're sort of trying to expand that initially just for colorectal cancer patients in Oxford, and then we'll see if we can expand it further in the cohort. But that would be another way of trying to take these slides which were made for clinical diagnosis. These are not research slides. These are things that were made for these patients to get diagnosed and treated. But then saying, can we use them to develop this really rich resource of digital pathology slides so that we can then integrate that with all of the other incredibly rich data in UK Biobank and correlate that with the germline genetics, with the proteomics, with the self-reported information, with the imaging and so on. So I think there is a huge potential for integrating clinical samples into UK Biobank, and we are trying to do that.
That was the coolest thing I've heard today. And if we have time, I'm going to ask you about machine learning on the digital pathology slides. But before we do that, I want to go to Alex and Yin, because actually they work at institutions in the United States that have their own biobanks. And those biobanks are more or less local to their institutions, as well as with a lot of public data sets. So I want to ask, maybe we'll start with Alex and then go to Yin. How do you think about taking these other large data sets and integrating across large data sets?
That's a great thought. And certainly within all of science, but particularly in epidemiology, anytime we make an observation, we feel a lot more confident in the observation if we're able to replicate it in one or more independent cohorts or independent data sets. And so we've tried to do that, to work not just with UK Biobank, but in other data sets like the All of Us project or large NIH-sponsored studies like the TopMed Consortium or TCGA, as you mentioned, to try and take some of the same pipelines that we're able to run in the RAP and deploy them in other cloud environments to make it easier on the people doing the programming, so they don't have to reinvent the wheel. And I think that's really a great opportunity with these cloud-based systems like the RAP, to not have to download the data, which would take many months, and also to be able to make it easier for the people who are actually hands on the keyboard doing the work.
I do want to come back to cloud-based systems in a minute if we have time. But Yin, how are you thinking about integrating across data sets?
Well, I think that will be our ultimate goal, but I think we are really at the initial stage, as Alex mentioned. I think it's more about trying to streamline the pipeline at this point. But down the road, I think with the more clear or straightforward data sharing policy, I think there will be a great step forward in terms of sharing these data and allowing people to use these data in a more broad scientific fashion and a broad community in terms of utilizing these data. But I think we are really at the early stage, and I think genomic data will be the initial, I would say, the pilot or the first to be along this line to be pushed forward. But other phenotype data, it's more challenging compared to genomic data because the way the data have been collected. But down the road, I see a great need or great demand in terms of trying to bring in rich phenotype data along with the genomic data or circulating marker data to really help advance our understanding of cancer and other disease in general. So I think bringing phenotype together cross data set will be a hard step, but definitely worth investing in the next several years to come.
Yeah, so maybe I can point this to Megan a little bit. Megan, as you know, and a lot of folks in the U.S. do not, Josh Denny from All of Us was at the last UKB Scientific Conference. So I'm wondering if you want to talk about some of the things that are going on between the UKB and other databases. Or if you can't talk about that or don't want to talk about that, you want to talk a little bit about the OMOP harmonization project from the UK Biobank, because I think that's going to be really central to what Yin is talking about in terms of phenotype harmonization.
Yes, so yes, UK Biobank data has now been OMOP'd, which is fairly massive, and I know they're busy, hugely busy, trying to get this data ready to be made available on the RAP. I am not yet aware of a release date. I'm not sure if our data analysts themselves are yet aware of a release date. But that will be coming soon, will be the OMOP'd version of UK Biobank. So yeah, another effectively a whole new data set, which is great, because it then means that any other data set that's been OMOP'd, they become harmonized together, so you can hopefully more easily directly compare various data fields, and for data replication and meta-analyzing your results, it becomes a lot more simpler. Yeah, so which is great.
I would like to make a point to the audience that I think we've had a seminal moment here where Megan Conroy has changed OMOP into a verb. So now things can be OMOP'd and we're OMOPing, which is very exciting. I mean, to me. So I hope the rest of you all heard that and recognize it. And I think Odyssey's going to appreciate that, seriously. All right, so if we can now go, tell you what, so that was really neat. I'm interested in how you all think that the cloud is helping with all of this, but maybe let's go to Kezia and talk a little bit about the digital pathology slides. And what I'm thinking about is how do you envision people applying deep learning or even very supervised learning to help sort of classification and diagnosis with those digital pathology slides?
So I think there's huge potential. And if we just look at other digital pathology projects that have already happened with collections of digital pathology slides, for example, from other cohorts or particularly from clinical trials, there's been a lot of really interesting work initially trying to get AI algorithms to sort of replicate our existing classifications. So partly replicating our sort of standard morphology, pink and purple H&E slide kind of classification, partly replicating the molecular classifications. And if any listeners are wanting to know whose names to stick into PubMed, Victor Kulzer and Jacob Cather, amongst many others, have done some really exciting work. I sort of hesitate to say that because there are so many other names on the papers, and they are sort of big, epic, multi-author collaborative projects. But those two authors will pick up a lot of the exciting papers. So there's already a lot of interest in sort of trying to say, can we replicate our existing classification but using AI? And then the next step is to say, are there other things that we can do using things like deep learning that don't just replicate a pathologist but go better? And so you might want to validate your images against survival, for example. Are there features in the digital pathology slides that are predictive of survival that go beyond what we already know about? So if you already have a pathologist to help you, you can look at things like stage or grade. So grade is how ugly the tumor looks, stage is how far is it spread. And we know those are predictive of survival in just about every cancer. And there is the sort of different histological types can be predictive, and certain molecular markers that we might get from immunohistochemistry, so antibodies that stain specific markers. So for those who are familiar with breast cancer, you'll have heard of ER-positive tumors, so tumors that express the estrogen receptor, and that tends to be predictive of the way the tumors behave and whether they respond to drugs that target estrogen, for example. And there are many other similar sort of relatively basic pathology things that we can do. And so the question is, can the AI go beyond that? And I think there's already exciting potential to look at that. But the difficulty is, do you have decent data sets? Because you need to have really rich, really complex data sets where you've got the digital pathology slides and the staging information about the tumor and the survival information. And then you might also want to account for potential confounders. So things like, age and socioeconomic status, and does the patient smoke? What's their blood pressure? Most of your other data sets won't have that information, but UK Biobank is amazing because it has all of the information you would want about the survival, but it also has all of the information you might want about potential confounders. And then if you want to ask questions like, are there specific risk factors that are associated with these tumors that have these features that the AI algorithm has detected? Well, you can look at that too. I should say we don't have all of these scan data sets yet on the scan slides. They're not going to be available for probably several years. This is going to take up the next few years of my life. But it's going to be really awesome when we do it.
That's awesome. Thank you so much. I have a question for Megan about somatic genomics data. But first, I would like to ask really sort of the clinicians in the room, Kezia and Alex. So obviously, reinforcement learning from human feedback is a really sort of hot topic in deep learning, as Kezia alluded to. And what I wonder is, so obviously that's happening right now within institutions. We know of hospitals where a lot of that's happening. But can you envision a world where that would happen like cross-institutionally? I mean, since we're talking about cloud and the potential of cloud for sort of revolutionizing some of this? And would you be willing? Let me ask a similar question. Would you be willing to participate in such a thing?
I can say that in the blood cancer space, we're actively working to do that, to start these kinds of collaborations. And what's actually pretty interesting about machine learning is that, whether it's in the cloud or happening elsewhere, is that, let's say I create a model, I have the ability to send that model to wherever the data is. And so even if one institution stores their data in the Microsoft cloud and a different institution stores their data in the Amazon cloud, or if someone's in DNA Nexus and someone else is in a different data enclave, the ability to port these algorithms in such a way that it doesn't really matter that all of these are sort of sealed off clinical enclaves that aren't is incredibly powerful. So basically, it's the whole paradigm of cloud computing is that you bring the user to the data rather than how I did it when I was in grad school, where we were downloading data to our local compute clusters. And so I'm really excited to see how this continues to evolve. And this is particularly helpful because with human data, there's all sorts of regulatory concerns, particularly clinical human data, where the data participants may not have been consented the same way that our UK Biobank participants were, with respect to data sharing.
Awesome. So I think I'm going to skip over, if it's all right, to Megan now and ask Megan. So we know, I can say I know from a DNA Nexus perspective that lots of people are using Mutek 2, both on CPU and GPU, on the UK Biobank data set. But are there any returns in the hopper coming back for somatic variants from the UKB? It's a audience question.
Not that I'm aware of. No, but there's a lot of data returns to be processed. So it's very possible. I mean, it's certainly something that, we will, if researchers are doing, then let us know, because it's something, we'd be very interested.
Take a tiny step
a small
back.
plug. We have somatic data, and others have as well, that I think we're trying to get returned. But the problem is that UK Biobank is so popular that the number of returns, I think, is like 6 to 18 months at this point.
Yeah.
Megan, if I could take a little step back for those who are in the audience who might not be super familiar with the returns process, could you just take like 45 seconds and just explain to people what that is?
Certainly. So when people sign the MTA, the contract to get UK Biobank data, one of the things that they say they will do is any data that they generate from UK Biobank will be returned to UK Biobank so that we can then make that available to researchers. And that's, so yes, anything that's generated, we would ask to be returned to us, and we have a process for that to be undertaken. And then we then clean that data up and then make it available to all the registered researchers so that they can then access that data. But we're a little bit overwhelmed, you might say, at the moment with just the sheer number of returns that we have. So there is a lag in getting that, we're receiving the data, but getting that data then up onto either Showcase or onto the RAP, depending on how we're making it available. There's a lag there at the moment. We do try to prioritize things that we think would be of most interest, but when we're receiving hundreds of returns a week, that becomes quite difficult to really, get on top of. So I'm not involved in that. It's the pure data analysts that do all of that. But yes, we have a lot, and there is a big time lag at the moment.
Thank you very much for explaining. And actually, I will tell you, I wasn't even quite aware of how big the volume of returns is. So that's awesome. I mean, that bodes super well for scientific collaboration, which is something I want to come back to in a minute. But first, I'd like to ask Yin. So Yin, you're very interested in early onset cancers, precancerous states. How would somatic returns help you in your research?
Oh, it definitely will be super helpful. I think it's one of the areas that has not been really looked into. As you know, that we looked into CH and lung cancer and finding a slightly strong signal for younger lung cancer cases between CH and lung cancer risk. And I think that's really one initial discovery we had, and potentially down the road, we are really interested in looking at how the somatic landscape potentially really affecting cancers at younger ages, because one possibility is that they have accelerated aging, and CH and other somatic markers may be relevant in this process. So that's really quite relevant to our research. It would be really nice to have some return data that we can really dig into.
Awesome. All right. So now what I'd like to do is ask sort of the last moderator question. And I'm going to ask each person this question. And then what I'd like to do is turn the Q&A over to the audience and ask them to put questions. So if you can be typing in your questions, I have one more moderator for the question for the panel. And that is, if you could wave a magic wand and ask the UK Biobank or the RAP, to do anything to help collaboration on the UK Biobank. I mean, we've talked a lot about collaboration. What would that be? Maybe we'll start this time. Actually, let's start at the top. Maybe we'll start with Kezia. Kezia, if you had a magic wand and you wanted to enhance collaboration on the UK Biobank RAP, what would you change?
I don't know. I think already one of the great things about UK Biobank is that there is this potential for collaboration, partly because there's this shared access to data, and that you can have so many different groups all looking at the same data at the same time, and just having that kind of immediacy of collaboration in a way that typically isn't possible. And yeah, previously, the one thing I kept asking UK Biobank to do was improve their pathology research. And then they asked me to help them do it. So I am not going to complain about anything else because they're letting me do that.
That's amazing. Thank you for doing that. Let's go in. Let's go next to Alex. Alex, what would you wave a magic wand and do?
So this is actually a little bit less related to the RAP and perhaps a little bit more related to the way that projects are set up in the UK Biobank. And so, with the RAP, theoretically, you can collaborate with any other UK Biobank, RAP-enabled user in theory. But in practice, we're actually limited by only collaborating with individuals who are listed as approved users on our particular project. And so you can add additional universities, let's say, to your project, but that involves sort of multi-university collaboration and collaborative agreements. And so one of the challenges actually goes back to this question about from the audience about returning certain data sets. So if the data set return process takes 6 to 18 months, then there's this gap where there's very interested researchers contacting me saying, I read this paper of yours, I really want to use this result that you generated. Can you send it to me? And the answer is absolutely not. That's a violation of the UK Biobank terms and conditions. And they say, well, can you add us as a collaborator on your project? And the answer is sure. Let's start the university contracting process, and that'll take another year because of the way that universities are used to working with each other. So if I had a magic wand to wave, I would say that rather than limiting collaboration to only people within approved projects, that there could perhaps be a mechanism where either certain data sets are sort of self-published within the RAP without being returned via the UK Biobank, so that I can make a file readable to others, or some other way of credentialing users so that users can work across approved projects without having to be added as an institution. I suspect that there's many legal and logistical hurdles that are far above my pay grade in making something like that happen. Has been a real challenge we've had.
Gotcha. I mean,
which is a good problem to have. I mean, it's great to have people want to collaborate with you across the world.
Yeah. There's a lot there. In introducing Yin, I will say that I am actually a collaborator on her project as a researcher there. So Yin, what do you think?
Oh, I think that's a tough question, but I agree with Alex. If there's anything that has been a challenge for us too, exactly as what he has described. So if there's anything that we can help, or there's a mechanism to speed up this additional data sharing process, that would be really helpful for everybody, for the scientific community, and also in terms of development in UK Biobank as well. So let us know if there's anything we can help in this process. I couldn't come up with a better idea in terms of collaboration. I think many people have utilized either the RAP or the UK Biobank in the to collaborate in an unprecedented way compared to other existing resources or open access data. So I think in that way, UK Biobank and the RAP is really leading the way in this front. So we really appreciate all the effort you guys have been put in this process. And that's my thought.
Awesome. I think that segues really well into that segues really well into Megan. Megan, any comments there?
Well, I was going to say very similar to what Alex and Yin had said, that actually being able to move the returns process onto the RAP so things become a lot more quicker would massively improve collaboration for being able to access that data and not have to worry about waiting or getting all the MTA signed, that because the data would be made available through each application individually.
Thanks. All right. I have a minor technical question. But any sort of general questions for the panelists before we sign off here? And I'd like to personally thank the panelists, both for being here and also for the fact that there was a list of somewhat pre-compiled questions, and I believe I asked them none of them. So thank you to all of them for responding to some very ad hoc questions. I personally really appreciate it, and I think it's been super interesting. I have certainly learned a lot. Let me thank the panelists again, and we'll go ahead and wrap up here. The recording of this, if your colleagues want to check it out, will be available on community.dnanexus.com in the next few days. Actually, there is one question from the audience for Megan, which I really want to hear and the answer to, and I get asked this a lot. Will there ever be tumor genomic data in the UK Biobank?
Well, I don't know. It's one of the things that we would like to see, but these sorts of linkages to, we wouldn't be able to access the tumors themselves. We would need to access any sequence data that has been undertaken on the tumors by the NHS. And those sorts of linkages take time, a lot of time. They're quite complicated. So it's something that's on our radar for, we would love to do it, but we can't make any guarantees if it will ever happen.
I think with that, we'll go ahead and sign off here. And if you're really interested in particular data types, come to the UKB Scientific Symposium in December, and you'll be able to talk to a lot of folks who run the UK Biobank and make your case for them. So thanks, everybody. Thank you once again to our panelists, and have a great rest of the day and a great weekend.